Electrocardiography is an extracellular recording. In case of extracellular recordings, the charges on the outer surface of the cells are important. So electrocardiography records the dipoles or the bioelectrical potential differences between the outer sides of two different groups of cells, muscle cells, in our heart. So let us have a look at these two different groups of cells. I have drawn two different groups of cells here. Down here I'm going to explain depolarization, and here we will see the recording of depolarization in electrocardiography. So we know that we have two electrodes. One is positive, the other one is negative or a neutral electrode. I have placed my positive electrode on the right side of these cells, and as the electrocardiography recordings videos proceed, you are going to see more often that we are really more interested on the location of the positive electrode. So when all areas are at resting membrane potential, outer sides of the cells are positively charged. All cells are positively charged on the outer side. Electrocardiography does not record any dipole or bioelectrical potential difference between these two areas, and so electrocardiography records a straight line. This horizontal line is called isoelectric line. Let's say the polarization starts on the left side. The cell group here become negatively charged on the outside. The cell group on the right side is still positively charged on the outside because the cells here are at the resting membrane potential. Now we have a dipole, and electrocardiography records this dipole or the bioelectrical potential difference. The question is, how will electrocardiography machine reflect this on the electrocardiography paper? Will there be an upward deflection and a positive wave to represent this dipole, or will there be a downward deflection from the isoelectric point and a negative wave to reflect this dipole? So we need to talk about one important generally accepted electrocardiography rule, and it says that if the positive electrode is on the positive side of the dipole, electrocardiography is going to record a positive wave. So the positive electrode is here on the right side according to you. The positive side of the dipole is close to our electrode, and during this depolarization, the electrocardiography will be drawing a positive wave, an upward deflection from the isoelectric line. Why did I put the same figure a second time? I want to imply that these areas are not really separate from each other in the heart. All areas in the heart are connected to each other. So it would be better if I drew the heart as a whole, because we know that our heart is actually working as a functional syncytium. If we repeat the depolarization now, when all the cells are positively charged outside, including the center area, there is no recording because there is no dipole. When a dipole forms starting from the left side, the middle and the right sides are still at the resting membrane potential, electrocardiography is going to make a positive recording. The depolarization will move to the central area. There is still a dipole here, and the positive electrode is still on the positive side of the dipole, and it's still recording a positive wave. And when all of the areas are depolarized, the outer surfaces of all of the cells in these areas will be negatively charged, and there will be no dipole, and electrocardiography, as you see here in red, is going to record nothing. Isoelectric line. To express this in some other sentences, you are going to see that this is also expressed as if the depolarization is moving towards our positive electrode, electrocardiography is going to draw a positive wave. We have talked about depolarization and how the electrocardiography waves form during depolarization. Let us move on to repolarization in the list here. The same group of cells are here. Our positive electrode is still on the right side of these cells. Before repolarization starts, all cells are depolarized. They are at the plateau phase of the action potential. So the outer surfaces of all of them are negatively charged. Because there is no dipole, the electrocardiography is going to record an isoelectric line. This isoelectric line is actually this isoelectric line at the end of depolarization. It is the line, isoelectric line between depolarization and repolarization. So let us say that repolarization starts from the right side. I know that you will immediately ask why I have picked the right side. There's a detail about this which I am going to explain when I talk about the T wave of electrocardiography. Please be patient with me. I request you to accept that it starts from the right side at the moment. So when repolarization starts from the right side, the cells in this group are going to return to the resting membrane potential, and the outer surfaces of them will be positively charged. The rest of the muscle is negatively charged. Our positive electrode is on the right side. According to you, it is close to the positive electrode, and so positive side of the dipole, and so the electrocardiography is going to record a positive wave. The repolarization will move towards the center. There will be still a dipole, and we will still have a positive recording. When all cells have repolarized and returned to the resting membrane potential, outer surfaces of all of them will be positively charged. There will be no electrical potential difference or dipole, and electrocardiography will again return to the horizontal isoelectric line. You may ask why I have drawn the positive wave of repolarization different from the positive wave of depolarization. This is again a detail that I am going to explain when I talk about the details of the T wave in electrocardiography. So here we can put it in another sentence. We can say that as repolarization is moving away from the positive electrode, we have a positive recording in electrocardiography. I explained depolarization and repolarization waves by putting the positive electrode on the right side. What is going to happen if I put it to the left side and the events are similar? Now, my positive electrode will be on the negative side of the dipole. If positive electrode is on the positive side, there will be a positive recording. If positive electrode is on the negative side, there is going to be a negative recording. And we can say that if the polarization is moving away from our positive electrode, we record a negative wave in electrocardiography. The same thing applies to repolarization. If I place my positive electrode this time on the left side, according to you, the positive electrode will be on the negative side of the dipole, and if positive electrode is on the negative side of the dipole, we record a negative wave in electrocardiography. Or we can express it in another sentence. If repolarization is moving towards the positive electrode, we we have a negative recording. In the last piece, I want to put the positive electrode at the center of the area. So, and examine what is happening during depolarization. At the beginning, resting membrane potential is present in all of the cells, and the outer surfaces of all of the cells are charged positively, and there is no dipole, and electrocardiography records a horizontal isoelectric line. If the polarization starts from the left side, a dipole is going to form because on the left side the cells at the polarized condition will be negatively charged on the outside. So our positive electrode is here. It is still, there's a dipole, it is on the positive side of the dipole, and it is going to make a positive recording. As the polarization moves toward the right, according to you, your positive electrode is still here. It will now be on the negative side of the dipole, and a negative electrocardiography wave will be recorded on the paper. When all of the areas are depolarized, outer surfaces will all be negatively charged, and there will be no dipole. Electrocardiography will again draw an isoelectric line. How can we express this in other words? We see here that first the polarization is approaching our positive electrode, and then it is moving away from our positive electrode. So I tried to make an introduction to how the waves are formed in electrocardiography in this video. Before finishing the video, I have to stress on a very important point. These cells that produce the dipole, either all of them are going to be in the atria or all of them are going to be in the ventricles. You cannot have some of them in the atria and some of them in the ventricles. For example, all the atrial muscle cells may be depolarized. All of the atria may be negatively charged on the outside, but ventricles may still be at resting membrane potential, so all of the cells there may be positively charged outside. You can think that atrial cells are negatively charged outside, with ventricular cells are positively charged on the outside, and there's a dipole between them. No, this is not possible because there is anatomically a fibrous tissue between atria and ventricles, and it acts like an insulator. It prevents the formation of an electrical dipole between the ventricles and atria. So the electrocardiography can measure the dipoles in the atria or in the ventricles. It cannot measure a dipole between atria and ventricles. So we have reached the end of the video about electrocardiography. Thank you for watching.